Good morning, everyone. Thank you, Sarajit, for organizing all that, and thank you to all the participants for taking time to be here. So basically, my understanding. So what I wanted to talk about is about technology, but more about also how we use this technology, which we originally developed, and then what the Molsoft continues in with such a spectacular success. But I want to say how we apply that to drug discovery and what are the new techniques and new challenges and kind of exciting new possibilities. Because as Sarajit already said, we started early, and it's not just that we were the first ones who developed the docking which actually worked, the flexible docking that worked, and that was the foundation of that was this internal cord mechanics. And then we proceeded to expand it to all kinds of different applications. And then all the docking methods which followed, they essentially copied our representation in internal coordinates for the molecules and our global sampling algorithm. That includes Glide does a very similar thing, but they of course they were later than us, and they used what we our descriptions to do that. And Vina as well, the Autodoc Vina is done by my junior colleague when I was at Scripps, so he would come to me, and then they also took the global optimizer from us. And we continue refining that. So basically, this talk is about, well, I call it leads from space, right? Leads meaning the drug leads. At the end of our procedure is a drug candidate, which then goes into trials. And I will show you the whole path from beginning to the end. And why from space? Well, is it from cosmos? No, that's from chemical space. And the space here is because over time, over these years, we moved from screening 100,000 compounds to millions of compounds, and now we're screening billions of compounds and possibly up to 1 trillion. So and at that size it becomes a space because it's not on your shelf. These are the compounds which can be synthesized, but they exist out there in the chemical space. So this slide also shows what are the essential parts of that. The one essential part is like those pictures you see. And by the way, every picture which you see here in my presentation, including except for the furniture, every molecular picture is done with ICM. So they basically open ICM. It's like your Microsoft Word for documents or whatever. And you can. It understands all the different objects. You can read them in. You can play with them. You can convert one to another, and you can create those live objects, and then you save them as a single document, and you send it to your colleague. They open, and you see all the views and all the particular details, both chemical and 3D and sequence and alignments, all in one document. So this all done with one. So when you see this picture, well, okay, let me create. Do you see my pointer, by the way? Probably not.
Yeah, you do.
Right. So because I was thinking if I can create a laser pointer with that. All right. So then the process is, of course, modeling, and then the hardware, which is not shown here, is your desktop, your laptop, your workstation, and then this is the front end, and this is the back end. Then there is a space, and then we define the problem. In this case, it's a particular protein, and then we look inside and we decide to target that particular location. Then we perform a screen. And that screen is just one of the data points, but we also, with Molsoft software, we can take your lead molecule and screen it against thousands of models for all kinds of characteristics, for activity against each of the other targets which your compound may accidentally hit. And as we know, all the small molecules will hit multiple targets. And then on the right, you see how do you define the project? You define the project by first you define you start from a disease in drug discovery. You need to understand what problem you are solving, and then you formulate the desired target activity profile, which may be quite sophisticated these days. For example, you may want to target a particular mutant, but not the wild type, and not the relative of that protein, which is very closely homologous, but otherwise you don't want to hit that other one. And so that's called the target profile. And target profile is more than just, okay, I'd like to hit this particular protein in this location. So it's more than that. It's all this definition. Then the Molsoft provides tools and compounds. And the tools and so here you see what the tools do. So all the modeling, and you start from is just finding which structures, what's the best model, what's the best collection of models which you want, where's the pocket, and then you perform different screens or interactive designs, which you can also do. A very popular Molsoft tool would be an interactive designer. Where you look inside the pocket and you start building your molecule chemically or modifying the existing molecule. And then scoring and so on. And then the big data, we need to continuously follow that stream, because as the new data emerge, then our methods should become better, because they should include all this data. And you'll see how we're updating our models in that regard. All right, so Andy will talk, who is next, will talk about the tools, and so this is just to show you that the Molsoft, this old Molsoft team, Max was my first, initially he was my grad student, and then he's a very strong scientist, and independent scientist, and also he's a lead scientist at Molsoft. All the initial ideas after a certain year, we developed together, and then Eugene is our key developer. Andy will speak next. Andy does everything, so he's an excellent modeler. He's a good scientist, and he also helps with support, and he's one of the reasons you get the reply in 1 hour or 3 hours, even at night of our time. Paulo was my former postdoc, is a fantastic scientist. Arman is a developer who does many things which nobody even know how they exist. So ICM, and again, I remember the pictures I produced was ICM, all of those, including the animated ones. Is portable, so it exists on all the platforms. You want to run it on Windows, fine. You want to run it on Mac, fine. You have a newest Mac with M4, fine. You want to run it on your phone, fine. You want to run it on the back end, on any Linux branch, it will run there. So, and the reason why we port it so easily is because it's a very tight code which the key developers know very well. We know every line, so we're capable of changing that, porting that, adjusting that to ever-changing computers and operating systems, and that's one of the very strong parts of ICM. It's not just done once by somebody, then these people disappeared and nobody knows how it works. It's not a collection of different things, but it's one tight, coherent body. So, yeah, so we started early. We developed this internal coordinate method, which then was the key breakthrough to allow sampling, which you can see here. The sampling gave us the global optimization. The global optimization gives you a set of solutions, and then we reevaluate those solutions and then rank them, and then we choose the best one, and that gives us this fantastic performance in docking. Now, the 4D thing here means that it's we can as a receptor confirmation, we can take multiple receptors which are slightly different, and some side chains may move, but we also can allow any flexibility around the dock, and it just costs you time. So the more flexibility you allow, the slower it becomes. All right. Let's go to the key data we use, of course, and you will use, and you are using already because you look like an experienced group of people. The first one is structure. That's our kind of home base, and we routinely chew through the entire PDB, and the PDB is, of course, a collection of well, that needs to be updated today. I'm not sure what the number is, but a few days ago or a few weeks ago, it was 226,000 files. These files are complex. You open some of the new files, they're gigantic files with many different proteins, and you need to make sense of them. Now, what is interesting about the PDB development is that it used to be mostly X-ray and a little bit of NMR. Now it's only 62% X-ray, 36% are cryo-EM structures. They grew very quickly. They are not as accurate as EM, but they give you a view into the complete structure. So if it's a large macromolecular complex, you will see the whole thing, and that gives you a perspective about what to target. If it's an X-ray, X-ray is still our preferred, of course, method because it gives us the best resolution. And for chemical discovery, you want the accurate definition of your pocket. The EM quality may not be enough, and definitely the AlphaFold quality will not be enough. So another particular feature of PDB is that it responds to the new challenges very quickly. When COVID emerged, just in 6 months, you see all the structures on that picture emerged. So people react very quickly, and then as a modeler, you should be able to quickly jump onto that and using those structures, find the new chemicals to solve the problem. Another source of data, you have the PDB derivatives, like, AlphaFold, you need to take with a grain of salt, in my humble opinion, because of these bubbles and things which AlphaFold does to you. We also use quite a lot the small molecule crystallography database, and this one is the open crystallography database, and we use it to derive the statistical or neural network based improved force fields based on statistics against that database. Now, other types of big data, the key data element would be the European Bioinformatics Institute products, and the central one is Campbell. Campbell is where I used to work in Heidelberg, that's why I like it so much, and the CH is chemistry, so chemistry, Campbell, and that's the Central Institute in the UK. You can see that it has lots of chemicals, but it also has the characteristics of those chemicals assays. And the reason I show that is, like in Molsoft models, we take all this data, and every new release we rebuild our models using this. What is the best model for log P? Well, we would go back and collect better data set. We'll clean it up, and then people at Molsoft will derive a new model. So that's a continuous process. That's why we release continuously new versions, because the data grow, and so with this data our models improve. Here is just the protein information in UniProt. And there are many interesting things there, like what are the mutations, how they associated with diseases, do we see that in structure? They don't necessarily follow every new structure released in the PDB, but we can do that with Molsoft tools. And then finally, the new chemistry was which moved from catalogs to so-called virtual libraries or virtual spaces, right? So this from billions to trillions, and that's where we have great hopes. And I will show you some examples to convince you that it will it makes a difference. Right. Unless you are specifically interested in AlphaFold, I will skip my kind of sniping comments about that. But if you read the AlphaFold and it shows you something like here in the middle, it doesn't mean it just means that it's a homology modeling tool. It just means that it doesn't know. Don't take this the bubbles as a prediction. It just says Well, I can only show you how very similar things look like, but if it's not far, I'm not a prediction tool. It's just the homology modeling tool. All right. Enough about AlphaFold. Let's go to our models. And Andy may show you a little bit more about this collection of models. But basically, the Molsoft software also comes with a large number of pre-built, quite sophisticated models. And the model will be based on a bunch of 3D structures, ligands, and then machine learning methods built on top of them, and then AI. And it's all taken from both 3D structures and 2D and the activity data, and allows you to produce an input. That's your trial molecule, which you maybe picked up from a screen, and then run it through all these multiple models and predict all kinds of activities of that model. So how it binds to EGFR, how it binds to JAK2, and then ALK kinase and c-KIT, and what is to your particular target, what's the docking score and the binding score? What are the physical and PK properties of the molecule? What's log D? What's the PK of different groups? Does it hit the hERG receptor, which is responsible for arrhythmias? So all these models so once you have a smaller number of hits, then you can characterize them as all these additional things. And these additional models are derived from this big data and continuously improved. So the full set of that, we call it Molscreen. The full set of that is we don't distribute because it's distribution becomes so big that it takes a long time to download. But we provide those services and we run it internally. But a certain number of key models are provided with the software. And the second thing here is to show you where this hit comes from, and it comes from screening of possibly up to trillions of molecules, but this chemical space is no longer just a list of individual molecules. It's a description of how they can be created. So they're called synthons and so-called reaction schemes, which then the software may combine into a molecule, and then it is screened and is checked for activity in your model. So here you have the lead generating model. You can produce new leads, so you can try them, or basically you search that space. You don't get out of that space, but then you find the molecules which are good candidates for the activity you're looking for. All right, that's the process. And then once you produce this list of actives or potential actives, then you send them here, and then you generate much larger information, predictive information about this active. You start asking all these other questions. All right, it hits my target, but does it hit the twin brother of my target? Does it hit the hERG? Does it hit the does it violate the fidelity of DNA duplication and so on? So all kinds of toxicities, all kinds of beneficial things will be checked here. So that's why I drew this arrow. All right. Now, one of the foundations of this set is the Pocketome. The Pocketome is a collection of pockets where we clean up all this quarter million PDB entries. We superimpose some of the structures or identical proteins by the pocket, just by the vicinity of the pocket. ICM has those tools where you can superimpose only the vicinity of the pocket. And then we derive the cloud of chemicals sitting there, and we also derive the cloud of pocket structures from that. And that goes into the models and training, and then we create one file, which then becomes a model, which contains all this information, as well as the algorithm, which needs to be applied to a chemical to predict its activity against that model. So the Pocketome becomes the foundation of the Molscreen, and there are different types of Molscreen models. There is no time to go through that. I don't know if Andy was planning to say a few more words about that. Now, So we are very thankful to the Novartis computational team, which for many years from early on worked with us, and we helped them to form their core software across all the sites of Novartis, not only Swiss but the American and abroad, and the Molsoft environment. They called it Focus because they put some of their modification of our interface, which Molsoft allows to do, so we can actually customize how the menus look like. They say, well, we never use this, but we use a lot of that, so can you add the menus here? So they made some changes, but essentially it's this the same ICM which you will be using, and then and this is a publication describing the success and statistics how the Molsoft chemist, biologist, and computational chemist. So it was used by medicinal chemists and biologists as well, and so that's actually most of the use. And that gives you this interface. You can read anything. You can memorize your project. You can build up all the data in a single document. It may have this alignments and the chemical tables. We were like the first to actually have the excellent chemical tables with all kinds of functionalities. And the 3D, of course, is there, and the views are stored. And then in that environment, you can actually create those project, predict the binding pockets, and this is the binding pocket prediction methods which we developed, which was then further improved at Merck by our users at the Merck company. So you may actually look into this paper. All right. Further, yes. Chemical spaces, Andy will talk about, but it's a separate discussion. But the bottom line is that there are two main things. One is the fixed catalog or collection where each molecule is enumerated. So it may be in the form of smiles, it may be in the form of SDF. That's a format, and the SDF can be two-dimensional or three-dimensional. But it also can be in the form of, we call it synthons and rules, and that's where you get access to much larger sets. And these are listed here: Wuxi Galaxy, Animine Real, Explore, Simplechem, and there are other ones. The chemists, as they learn how to do that, they join the team, and we help them to actually represent their chemistry. So we helped Wuxi to represent the chemistry in the compact form that gave them access to bigger spaces. But of course, the bread and butter is your collection, or maybe your favorite chemical vendor, or maybe your favorite chemist. If your favorite chemist will give you the building blocks and rules, you can, with Molsoft software, you can actually build those collections, save them, and screen them. All right, these are the chemical spaces. This is the representation of those chemical spaces. This is William Shakespeare saying to T or not to T. T meaning trillions. And that's a new challenge, and you may be bold and decide to screen trillions, but even in thousands, you can find your happy lead. So that's an option. All right, now to mention quickly that the biggest acceleration, also to show you that we continuously improve. When the GPU craze started, we were the first to actually port this sophisticated docking algorithm. So before us, the MD was ported to GPUs. That was done with Amber, and there was some improvement there, some acceleration. But here we achieved a dramatic acceleration of at least 1,000-fold, maybe 10,000-fold, when the GPU NVIDIA engines would allow us to screen, to dock 100 molecules per second. The previous, even the best form of ICM, which was better than other software, was in a couple minutes, let's say, for a medium-sized compound. But here it's just really dramatic change, and then there is a particular different engine, the GPU-based engine for that. And then for that extra fast and extra large throughput searches, these are the new tools. One is called Ginger to generate compounds, low energy confirmations of the compound. And then Reach is the fast docking, and the right is docking to this pharmacophoric density. This is to search for isosteres. And then there are different iterative engines where you, and the Combi Reach is one of the latest ones. So these are mostly services. You don't need to. So once you prepare everything, your project, and you have the initial hits, so then you can go there, and then also it will be can take your project and then basically do that as a service as well. So because it's a heavy machine here. But if you some companies get it and use it as a discovery engine, so started doing that, and NIH, for example, here in the United States, started using that, and it's very exciting for us because we help them, we work with them, we collaborate. All right. Now examples. So I have probably need to wrap up. A few examples of projects. So this is the projects of Alzheimer's disease. So the problem here is so-called hyperphosphorylated tau protein. That's a collaboration with Andrea Cavalli, who was in my lab for sabbatical. Stefano De Muro was the key chemist there. And what we wanted to do is to find a single molecule which hits 3 kinases which phosphorylate tau. And we managed to do that. And quite so only with Molsoft software screens. So you're screening against 3. So it's a challenging problem because how do you hit 3 different kinases but not much more with a single compound? That requires a lot of modeling. So now there is a follow-up paper just submitted with an improved version of these molecules. So we continue working with them. So you can see what was achieved. So there's 3 activities. They're all in nanomolar range. This is under 100 for Fin, and then DIRC1 alpha, GSK3 beta, and Fin. All right. So now this is a project which is currently in progress. It's a huge challenge. The challenge is the viral diseases, all these different viral diseases for which they're mosquito borne diseases, and these viruses are quite bad, and these viruses are called flaviviruses, flavivirida, Zika, dengue, West Nile, Japanese encephalitis, yellow fever, and previously, and there is these viruses have a protease, which helps to mature the virus, and people tried the main binding site, which is here under this blue blob. There is a binding site. But we realized that this binding site is not very good. It's flat, it's charged, and we started looking for allosteric sites, and we also started looking for different conformational states. We found them, and we with ICM we docked and screened and found the first-in-class inhibitor of this allosteric states, one of the allosteric transitional states, which will stabilize the protease in inactive form, and that's an active process that shows you this NS2B wrapping around the NS3. This is the protease, and this right next to the protease there is a helicase, and there so we can, and also generated with ICM, by the way. So here you can actually produce an alignment. You can look for how conserved is the pocket between the different viruses, as well as within the variation of the same virus, right? Because we have multiple sequences for the same virus. All right, now cancer. So I'm only talking about very challenging projects which we're doing in the lab. So I'm not at this point including the projects we do in collaboration with other startups or big pharma. So this one, we have a colleague at the Morse Cancer Center at UCSD, and then Jason Siklik, and this is Dionysius Siegel, who is our chemist, and Adrian Sanchez Fernandez is who worked experimentally on this project. So what's the target here? The target is this huge sausage here, which is called UNC80, and what is about it is that people notice that in some gastric cancers, UNC80 is overexpressed in the cancer cells. And they told us, can you do something about it? It's not an enzyme. It's a very large 1,000 amino acid structure that has lots of pockets. We did the pocket prediction. We found one pocket at the interface with this sodium channel. We targeted that. We selected only 6 molecules, or 8, I don't know, I think 8, yeah. And then 2 of them were active, and they showed that they're specific to this mutants which we studied. And so that now continues in Jason's lab. It was a spectacular finding because it's no one even approached vaguely that structure because it's so big and so it's so not obvious what is the active site of action is, but the tools allow us to do that. Now this is another challenging problem. And challenging target, and it's called GNAS2. This is G-alpha protein, G-alpha S protein. That's the one which works with GPCRs. And there's a particular mutant, which is the R, meaning arginine 2-1 replaced by C, which drives several cancers. So we build the model. Here you see in yellow, that's your cysteine, which replaced the arginine. And sitting right next to ADP. And we used Combridge, and we screened billions of compounds, and we were curious about whether we can fill that density. You see, that's a predicted pocket by ACM. And then, sure enough, we found some candidates. Now they're being tested, and we have 2 types of candidates: the ones which are covalent. It means that they will actually covalently connect with the cysteine, and the ones which are not covalent. Because there's another mutant which is R201H, which is also is of interest. Now this one, I just got the good news only a few days ago. In cancer, they're checkpoint inhibitors. What do? They go and look at the interface between cancer cells and immune cells, CD8 T cells. And at this interface, there's a PD1 on the immune cell side and PDL1 on the cancer side. And they're called breakpoints. So they say to the immune system, don't kill me, don't touch me. We want to break that interaction. So antibodies do that. So here the antibodies, nivolumab, pembrolizumab, and the ones which go to the PDL1 side, which is on the cancer cell, are those durvalumab, avelumab, and so on. Now, antibodies are great, but we wanted to find a small molecule to do the same job. And there was a discovery that if you promote dimerization of PDL1, it leads to internalization of that and disappearance of PDL1 from the surface. And you can see here the view from the top. Molecules which were found were too big and they were not good. So then we've done this screen with ICM tools, a large screen, and we found much more drug-like molecules. And here you can see the view from the side. It's basically a channel between 2 subunits. All of that, of course, is ICM, what you see, the pictures and the screen as well. And now we got 2 molecules which are active in that assay. That is the published one, so that's not ours. That's this long molecule, just a view from this side if you look from the bottom. But we found much more drug-like molecules here. This is another active project, another Dawkins screen, and then Combridge also participated, but ICM VLS, the one our mainstream tool was used to redock and improve the main hits. And here we inhibit the protease from the proteasome of Trichomonas vaginalis. So the requirement was not to hit the human proteasome, even though some cancer drugs want to hit human proteasome, but it's dangerous. They're toxic. But we want to hit only proteasome of Trichomonas, but not of human. And so you can see the kind of hits we found. I cannot show you the chemical structure because some patenting will be going on soon. Now we also the Molsoft and Andy participates, and all of them participates in many biotech startup collaborations. They basically come to us and they can say, can you help us with the fast discovery of some leads? And we agree. And just to show you how quickly it can be done, this is a collaboration with a company called Ailean. And in just 6 months, we were able to generate a better version of venetoclax. For the cases, venetoclax is the molecule, is a popular cancer drug, which binds to BCL2 and BCL2 blocks apoptosis. This drug activates apoptosis, and the cancer cells start dying. But the problem was that over time, some resistance developed to the venetoclax, and the new ideas were necessary. We found a new one, and just the whole process was 3 years with the computational side, and the first testing was just 6 months. Now it's all patented and in clinical trials, so it's no longer waiting for clinical trials. You see what we've done? We identified a different pocket slightly lower. It overlaps with the middle part. Found a different molecule which takes that pocket, can hit the venetoclax resistant mutants, and otherwise has good properties. Has better blood properties as well, and less side effects. All right. Another big success was also with collaboration with Ailean was the menin MLL focus. The first drug against that was just approved, but they cannot deal with mutants, and we were looking at the mutants of that situation. That's the menin, and menin in some cancers, the partner of menin, which is MLL, and this is a little fragment from MLL, binds to this menin. Menin is in blue and MLL in green. Now the requirement was to find something which will basically make it impossible for MLL to bind here. But the problem was that the pocket is wide and deep, and there are also mutants resistant to the just the emerged first one. And so we found another solution where the compound goes different way, and using the same tools. Now, again, this is also in clinical trials. The name of that is we call it balaminib, because the initially company was called Bala, and then they was moved to menin. So balaminib now is approved for clinical trial. It's actually already, I think it's past the phase 1. All right, now the third success was Fleet 3 mutant. So here it's a long story. I will not tell everything, but basically this Fleet 3 receptor has a kinase, and this kinase becomes constitutively active with some mutations in this part of the protein, and then you want to inhibit that. There's also a particular mutation, and then there are several domains here, and we performed a relatively small screen, 10 million. Well, it's still a huge screen, but that was enough to identify a small number of hits, and then from that 3 series emerged, and they led to the successful candidate which went to clinical trial. This to show you the pocket and the candidate. Well, the candidate again. So it's the we call it a skin. So like we put something on top of the molecule so the chemical structure is not obvious. And this is called lamotinib now, and it's in phase 1B. And you can see that it has fantastic characteristics in terms of the nanomolar IC50, like 0.36. Come on, that's 4 nanomole. And that was in a very short period of time. Now, this is another interaction, but the reason I show that shows that not only chemical structures you can screen, but you can also have the peptide-based constructs, and you can, with ICM, and Max is the key developer of those scripts, and he works with Fog Pharma. Donovan Chin is the VP of data science there, and it can actually find unusual, unnatural amino acids here, and the design that's staple as well, and they call it Helicon, that construct, and that's the key product now of this successful company called Fog Pharma. Look it up. So thank you. The final thoughts: ICM provides a modern front and back end environment, like basically comprehensive environment to play with chemical biology or discovering or optimizing or designing. I didn't, talk or show the things like ligand editor and so on, to do different things and go from just a vague definition of what you want to do to actual small molecule or peptide, which does that. And then I also told you about this new horizons with synthons and blocks and the chemical spaces, the new hardware which enables dramatic acceleration of this to be able to search those chemical spaces, and then the other types of data and the growth and our ability with scripts. ICM is a scripting language. You can actually write scripts just like you do in Python. You can mix it with Python, but there's its own language which is kind of similar, a scripting language. So you can script all kinds of large-scale analysis of databases. And then the 3 examples which we did in collaboration was an illustration how quickly you can go to drug discovery. It's easier for me to show that because ICM also was used in some big companies, and there it was an integral part of almost any new drug because everyone used this environment. And ultimately, that as a computational scientist, make sure that you work with chemists and biologists and disease models to translate your findings into a candidate. These are obvious things. And I thank to all these people with whom we worked for this particular project. We of course work on other things, and this is our only marketing effort here. Even though this is for some other company for pest control, I forgot what that is. I think ICM is also Indian composer. No, something. I forgot. Once I looked up what ICM is, and there's some other interpretation of this abbreviation. But for us, it's internal coordinate mechanics. And yeah, thank you for your attention.